Okay, this is an introduction to lecture 3. I probably, based on how long it's taken me to make these, probably won't be doing these for every lecture, but I do think at the beginning of the semester, we're introducing a lot of basic concepts that are going to be important throughout the whole semester. So if you get a good feel for these ideas, it should make your progress through the course a bit easier because you won't have to be relearning all the topics as we go through. Everything I'm talking about in these first 4 lectures is you're going to be seeing examples of these concepts over and over and over again in different organisms with maybe slightly different genes or slightly different functions for those genes, but the same patterns. And so the ones we're going to look at this time are sort of 5 basic concepts of sort of how embryos go from single cell to, in the case of humans, the baby, or in case of cats, the kittens that are born, in case of frogs, the tadpoles, and so on. We'll look at sort of what cell fate means, and we'll talk about differential gene expression, and you may want to review what I talked about when I talked about genes, like how, what we mean when we talk about gene expression. And we'll kind of build on that in the next lecture. The idea of combinatorial control, so the idea that it's not just RNA polymerase that transcribes genes. There are a series of special proteins that determine which genes are on when in the organism and where, and how a whole set of genes that are needed for a function, like forming muscle or forming nervous cells or something, are turned on at the same time. And then we'll talk about the terms specification, determination, and differentiation. Again, we'll revisit those terms multiple times through the semester. Okay, long introduction, but I hope the video itself won't be too long. So the first basic concept in development is cell division. So you start with let me just grab the pen here. We'll do purple. You start with your fertilized egg, which is a single cell, and then you go through divisions. And so the first division leads you to what is called the 2-cell stage, the second division, the 4-cell stage, and the divisions are all coordinated. So, the 1 cell divides, and then the second division is usually at the same time between the cells. And so you've now got 4 cells, and then there's an additional, I don't even know where the plane of division is here, but you've got 8 cells and then additional cells. And so one thing to notice is in embryos, you want to develop rapidly because the embryo stage is usually very vulnerable. It can't swim away from a predator or run away, and so you want that very rapid development. So what happens is the cells divide very, very quickly, but there's no growth in between. So notice that this stage, which is quite a bit later, it's the blastula stage, which we'll talk about momentarily. It's almost the same size as the egg. And that's because the cells, as I said, they divide without any growth. So the cells are smaller and smaller and smaller, but the organism is the same size. A zygote is the term for the fertilized egg. So the fertilized egg, it's a single cell. Zygote is the correct name. It will go through 1, 2, and 8 cell stages. Around the 8 to 16 cell stage, it forms a solid ball of cells that is referred to as the morula. And then during the next couple of divisions, there is a process by which a cavity is formed inside. So the blastula is a hollow ball of cells. And this, it's probably not super clear here, but if you take a slice through this stage, then you look inside and you see that there's this cavity inside. It's like, think about a tennis ball. The inside is just space and there's just the cells around the outside. And those are going to be important terms. We're going to talk about them repeatedly when we look at the development of different organisms. The second one is pattern formation. Pattern formation is a general term for how a particular body plan develops. And it can be specific to a particular tissue, it can be specific to the whole entire organism, etc. So pattern formation is the different positions along the body, along the developing embryo, and that somehow each cell in the developing, so here in the blastula, each cell knows approximately where it is in the organism and has some information about what tissues it's going to become. Okay, so that's pattern formation, and then positional information is how that's determined based on where, in the organism the cell is. It has the capacity to form part of the whole pattern, which is, an eye and brain at the front end, an ear at the front. The belly is different from the back, the tail is different from the head, and so on. And so you have, that's what pattern refers to. And we'll later in the semester, we'll look at sort of different hypotheses for how different gradients of gene expression lead to these, different fates in the cells and so on. So things to know about right now are body axes. So there's the anterior posterior axis. So if you were thinking of a cat or an elephant, for example, the head is the anterior end and the tail is the posterior end. And then there's the dorsal ventral axis, which the dorsal is the back and the ventral is the belly. And then there's left and right axis. And for organisms like us who are bilaterally symmetrical, we have a left and right side. And we probably won't talk very much about the left-right specification. We'll spend more time talking about dorsal ventral and anterior posterior. Not that the left-right isn't really interesting, it kind of is. And you can certainly look that up yourself if you're interested. And then polarity just refers to the fact that, one end of an organism, one side of an organism is different from the other. Okay, so when we're talking about polarity, we're talking, if you think about DNA, for example, one end is the 5' end, the other end is the 3' end. They are chemically different from each other, so that's polarity. In the case of the organism, though, we're talking about, the anterior pole of an embryo versus the posterior, and so on. And they are different. That means different genes are expressing, the tissues look different, the cells are maybe behaving differently, and that sort of thing. And then the germ layers. So we'll be talking about the germ layers more, but when we look at an early developing embryo, the outside is called the ectoderm. This is very early in development. So ecto means out. There is often tissue inside, which is endoderm. Endo means inside. And then there's the tissue in the middle, which is the mesoderm. And we will talk about this in detail in the actual lecture, but just be aware that the germ layers refer to the organization of a layer of cells that's going to eventually form the outside of your body plan, ectoderm, with some of it forming the nervous system as well. The mesoderm, which is going to form muscle and a lot of your internal organs, and then the endoderm, which forms the gut and the lungs and the digestive tract, that sort of thing. The liver, I think, is also endodermal. And so these different layers have already early in the development, when you look at a cross section and you can see these layers of tissue, each of the cells in different areas have different fates. And we'll spend a bit of time talking about these germ layers and what kinds of tissues derive from them. Okay, and then morphogenesis refers to major changes in form. So this occurs, so for example, this is the blastula this looks like a sea urchin to me. It is a sea urchin, yes. And basically from the blastula stage, which is a hollow ball of cells, you have certain cells that migrate inside. You have gastrulation, which is like a little invagination, followed by active migration of the cells inside to form a very different looking structure here, with certain cells doing one job, other cells doing a different job, and then the cells on the outside, of course, The cells move around and rearrange until you end up with um, a pluteus larva. So sea urchins have a free swimming stage before they settle down and become sea urchins as you know them. And they've got all these different tissues, the stomach versus the esophagus, the intestine. There's these spicules that give it structure and so on. So you go from one form to another. That is what morphogenesis is. And it involves the rearrangement of cells. So cells move to different locations. These guys here, for example, are actively migrating to move everything inward. Gastrulation, which we'll spend some time talking about, where the certain of the cells on the outside move to the inside and they're going to form those endodermal type structures, and then migration. So we'll have examples of all of these in upcoming lectures. And then cell differentiation. So cells go from being not specified at all to actually adopting their role as a liver cell or a part of the brain or something like that. So they go from being undifferentiated, having the ability to form either all or many of the different kinds of cells that an organism has, right through to the differentiation stage where the cell is now become whatever it's supposed to be, right? It's a skin cell on the tip of the finger. So that's the role that it's going to play. And then finally, growth. So early stages of embryos, the very early stages, there's lots of division, but not much growth. But then as the organs start to form and the stage, the embryo goes to the later stages, you will see lots and lots of growth, of course. Think of pregnancy and how big women often are before the baby is finally born. And then also the idea that when the body is being formed, there are different stages that grow at different tissues that grow at different rates, right? And so this is just one example, is that it leads to a real change in proportions. So that, for example, at about the 8-week stage, the baby is the embryo is very, very tiny, but something like a third of the body is head, right? At the 12-week stage, there's already somewhat difference. The body is grown more. It's probably it's almost a quarter of the body size, and then at 16 weeks. Again, the head is proportionately smaller than it was. And finally, when the baby is born. Now, the head of a newborn baby is still a remarkably large proportion of the total body size. Think about babies versus adults. Babies and toddlers. One of the reasons why toddlers tend to fall and bump their heads a lot is that they're a little top heavy, right? The head is proportionately larger compared to the body than it is as they grow older. And so their center of gravity changes. And so we're not going to there's interesting examples of how certain kinds of organs and so on develop, and there is differential growth as part of how that organ takes shape and its eventual form. But we don't have time to cover everything in the class, and so I've opted not to spend much time on this. If there was ever an advanced course that spent more time on the organ part, the development of different organs and so on, then we would spend more time on that. Okay, and so then another major concept, those are the 5 kind of basic concepts, but there's some other major ones that we're going to look at. And one of them is differential gene expression. And you will see examples of this repeatedly over and over. It's one of the main ways that, the most important way that a fertilized egg becomes something like your tadpole. And that is because after cell division, the first few cells that are formed, each of them then has certain genes that are turned on and others that are turned off. So some genes are activated that are required for a particular function. Others are shut off because they're not needed in that cell type. And that leads to, here's I don't know, this is about a 32 cell stage embryo, and it basically has different cells that have different fates. So these cells here have one set of fates. These cells might have another fate, and these cells might have yet another fate. And they're not done becoming, in this case, ectoderm, mesoderm, and endoderm, which we'll, as I said, we'll talk about more in the actual lecture. But they're on their way to forming those particular types of tissues. And then as development proceeds, there will be additional sets of genes turned on and off as the ectoderm is divided into either skin or nervous tissue. So those are two different kinds of structures that are formed by these types of cells, and the specific genes for each are going to differ. And so as you go, every time the cells divide or rearrange or any kind of change takes place, you're looking at a different batch of genes that have been either activated or turned off, or most of the time some on and some off, that will then restrict what a cell can be. So at the beginning, the cell could form any structure, but after a few divisions, usually the fates are somewhat limited. So, one of the 4 cells can only form a certain subset of structures, and the same with any of the others. Each of them still has the capacity to form lots of different kinds of tissues, but it gets more and more limited. Think of going through a maze where every time you get to a point, you can turn right or left, but once you've turned left, then there's only a few, there's the decisions that follow are limited, as opposed to when you're starting the maze and you could end up anywhere, right? So I don't know if that's a helpful analogy or not. Somebody can let me know. And if you can think of a better analogy, then please do share it with the class. I'll put up a discussion page for people to share their ideas. Okay. So the idea is for cells to become more and more specialized, and when you get to this is the tail bud stage of a frog or a salamander embryo, and at this point, most of the cells have adopted just a single fate. They are part of the gills, the stomach, the intestines, the skin, the muscles, the heart, etc. And so they've adopted whatever the fate is that they're going to have in the overall embryo. So, and I will briefly touch on combinatorial control. Combinatorial control refers to the fact that it is not just one protein, an RNA polymerase, that initiates transcription, is that you have a whole set of regulatory proteins. And so you might have a transcription factor like W here, which is just a made-up example, just a general example. And it's possible that when protein D is also present and bound to it, it acts as a transcriptional activator. I'm sure you remember from other classes that this arrow is how we show that the gene is being transcribed. And so therefore this gene, suppose this is gene A and this is gene B, or tissue A, actually it should be, that's not right. Let's just fix that right away. This is going to be gene A2. But this is going to be in one tissue like muscle. And this is going to be like nerve cells or something. Nerve and muscle are so different that it's easy for us to use those as examples, because you can kind of picture the cells and feel automatically that they're very different from one another. So in this case, this gene is active because an activator protein is present, and that protein interacts with other regulatory proteins, as well as, say, RNA polymerase that's sitting here in the regulatory part of the gene, and that leads to the transcription. However, in another cell type like muscle, the D protein is not being expressed, but the G protein is, and G is what we call a repressor. This is the symbol for repression. And so the repressor protein is preventing expression of gene A. Why? Because it's required for the function of nerves, and in the muscle tissue itself, it's not needed, so it's kept off. And combinatorial control is, if you look at the nerve cells, there's a certain bunch of transcriptional activators and repressors that are present, and they are responsible for the expression of all the genes that are needed for nerve cell function, but not no expression of the genes that are needed for other cell types. And then the opposite is true for the muscles. So the muscles have also a certain set of activators that turns on genes specific for muscles, and a certain set of repressors that keeps the other ones off. The body is very efficient. There's no value to expressing proteins that are not needed in a particular cell type. It takes a lot of energy to transcribe and translate genes, and so therefore you don't want to be expressing anything that you don't need. And then coordinate control is a subset. It's when the particular, so this is, cell type A, and maybe there's a whole set of genes that are required for this cell type, and you want to turn them all on at the same time. And so you have the same regulatory protein. In this case, it's B that binds to the W region, the W protein, and causes the activation of the genes. And when we see this kind of coordinate control, you're turning a whole bunch of genes on at the same time that all have a similar function. They're related to a similar function. And the key is that they have the same sequences upstream that your regulatory proteins will bind to. So these could be scattered all over the genome. You've got, maybe there's 10 different ones, and they're all on different chromosomes and so on. But if they have the same regulatory sequence, then these two proteins are able to combine and turn on the genes all over the genome. And hopefully that's a clear concept. Okay, and then the final thing I just want to briefly mention, and we'll talk more about in the class, is specification, which happens before determination, which then happens before differentiation. And these are things that have been defined based on the results of experiments, and we'll talk a lot about those experiments. So cells that are specified, quote unquote, know what they're supposed to become, but it's a reversible process. So what that means is, in terms of thinking of the gene expression, some of the genes that are specific for a certain fate have been turned on. And if left undisturbed, they will proceed to form the structure, say a muscle cell, something like that. But it is possible to reverse the process if new information is received, if new signals are received. So think about two things. A cell that is specified, if you were to take a clump of those cells and then just culture it separately and then observe what happens to it on its own, it will progress and form the structures, the type of cells it's supposed to form. And you'll see examples, especially in some of the early stages of embryogenesis, of these kinds of experiments, because this is some of the first information we had about how these processes take place. But the process is reversible, not under natural conditions, but in terms of experimental manipulations. You can change what the fate of those cells will be, and we'll talk about that in the class. Determination is the next step. So at this point, there are additional genes turned on and others that have been turned off, but these are now not reversible. So the difference here is that the process can be reversed. So the cell, if you leave it alone, knows what it's, quote unquote, knows what it's supposed to be. It's expressing the genes that are needed, and if left undisturbed, will go ahead and form whatever that structure or whatever that tissue is. But if something changes, you can reverse it. When cells are determined, even if you move them to a different location, provide them different signals, they will not change their fate. The changes that have taken place are what we would call irreversible. And then finally, differentiation is when that cell has adopted its fate. So it becomes a functioning nerve cell or muscle cell or skin cell or whatever it is that it's supposed to become. Okay, I hope this was helpful. It would be good if someone would let me know if you know, after a couple of lectures, if this sort of introduction is useful. Because if it is, I'll make an effort to really try to do more of them. Even if, as I said, I can't probably do them for every single lecture. But if you find that it's not really that helpful, it's kind of a waste of my time to do it. So if a lot of people don't like these or don't find them of value, then I could try to provide other kinds of resources to help you prepare.